A groundbreaking shift is underway in the world of computing. Melbourne-based Cortical Labs has introduced CL1, the world's first commercial biological computer running on living human neurons. Unlike traditional AI, which relies on silicon and artificial neural networks, CL1 integrates real human-derived neurons onto a computer chip, creating what they call synthetic biological intelligence. By transforming blood or skin cells into stem cells and then into brain cells, Cortical Labs cultivates living neural networks on specially designed silicon substrates. This blend of biology and electronics marks a whole new approach to information processing. Central to the CL1's design is a micro-life support system that keeps the neurons alive and functioning. This system resembles a miniaturized circulatory setup. Pumps emulate a heart, fluid reservoirs supply nutrients like glucose and amino acids, and filters remove metabolic waste akin to kidney function. A gas mixing unit sustains a balanced oxygen-carbon dioxide-nitrogen atmosphere. Temperature control maintains a consistent 36 to 37 degrees Celsius environment. The result is a self-contained habitat where brain cells can live, connect, and compute. Once the neurons establish themselves upon the silicon, they form synaptic networks that actively process signals. Thousands of microelectrodes embedded within the chip record electrical activity and feed data into classical hardware systems. Remarkably, CL1 can learn and adapt in real time, with higher speed, substantially lower energy consumption, and far less data than large-scale digital neural networks. Rather than merely mimicking biology, this system harnesses its real power. The immediate potential of CL1 lies in life sciences. Personalized medicine is a primary frontier. For example, consider patients with epilepsy or Alzheimer's disease. A personalized CL1 module could be seeded with neurons derived from that individual's cells. Researchers could then test which drugs alter neuronal activity most effectively, thus tailoring treatments at a cellular level. Circling away from generic, one-size-fits-all medications, treatments could become precise, more effective, and potentially safer. Beyond healthcare, the long-term vision expands into a reimagined AI paradigm. We're used to machines processing voxels of data on enormous clusters. CL1 invites us to envision devices that compute using inherently efficient biological substrates, operating with speed, adaptability, and energy minimalism. Picture cell-powered processors that sense and respond to environmental changes in a more natural, organic, even lifelike manner. Such systems could underpin next-generation robotics, environmental sensors, autonomous scientific exploration, and possibly forms of intelligence that don't rely on vast arrays of digital transistors. Currently, CL1 is geared for laboratories and research institutes. Each unit is custom-built, tailored with the lab's own cultured cells. Cortical Labs plans to ship the first systems later this year. As CL1 enters real-world testing, we're likely to see collaborations with universities, biotech firms, and pharmaceutical companies, potentially giving rise to a new field: biocomputational neuroscience. But while the science is thrilling, CL1 raises important philosophical and ethical questions. First, what is intelligence? If living human neurons in a lab can process information, form connections, and respond to stimuli, is that intelligence or even life? Until now, intelligence has been associated with human and animal brains or with artificial programs. CL1 blurs these boundaries. As these neural networks evolve, do they deserve moral consideration? How do we monitor and regulate a system that combines life with machinery? Secondly, the user-machine relationship could shift dramatically. In traditional AI applications, we are active agents, machines assist. With CL1, we might take on the role of observers or facilitators, watching biological intelligence behave, perhaps even adapt in ways we didn't explicitly program. We might ask, who is performing the computation, the human designers or the living cells themselves? Thirdly, There's a profound ontological shift. We've long viewed nature as separate from technology, something to be harnessed or mimicked. But CL1 demonstrates a synthesis. Technology built with living matter. It's no longer simple biomimicry. We are embedding nature into machines. Could this herald a new era where human-made systems are hybrids, containing life forms, processing information, and responding biologically to their environments? If so, where do we draw lines between biology, machines, and life? This technological turn-hop invites a broader reconsideration of our role in intelligence. We don't merely design systems that think faster or more efficiently.